Thank you, everyone. And it's already been mentioned, I'm representing EuropaBio and EBE, and as discussed this morning who that represents. So instead of restate that, maybe I'll be the first to say Happy Halloween. And thank you to EMA for hosting this constructive workshop. So I'll try to briefly address the three topics that we've been asked to address, give some background of our thinking, and some of it is actually suggesting back to understand more of what the EMA's thoughts might be and putting those thoughts into the guidance. So specific to the concept of a stepwise approach for non-clinical development, drug development is generally stepwise, whether it's an innovative molecule or a biosimilar. So we certainly agree with the outline that was proposed in the draft guidance. Obviously, we do understand the target, understand the space we're working in, understand the characteristics of the molecule, the biologic characterization of the molecule. Before we'd consider in vivo studies, we then would design the in vivo studies before going to clinical studies. The difference for biosimilars is that the determination or assessment for in vivo studies is entirely by the sponsor and presumed to not be needed unless there are some differences that have been identified in the non-clinical or pharmacologic, meaning biologic characterization of the biosimilar compared to the reference product. So there's the main departure, I'd say, in the non-clinical aspects is that the biosimilar guideline infers or implies that in vivo studies are probably not needed, depending on how close the molecule matches that of the reference product. And we actually agree with that. But of note that determination of how close the molecule is to the reference product is being made solely by the sponsor, not with any regulatory input at that point. So we agree with the outline. It's logical, it's scientifically sound. But then we get into the aspects of what the non-clinical stepwise approach tells us as we might approach the clinical aspects of development. And that's where things might differ or get be enhanced. Because the appropriateness of the clinical trials based on the non-clinical and pharmacologic data is really still being assessed by the sponsor. The sponsor then approaches each of the national authorities through the trial application process, and they evaluate the protocol and the available data, probably more for safety in some aspects, country by country, for the similarity, let's say, of the non-clinical and the data that's presented. So there, as we discussed this morning, there is no mechanism to have a regulatory assessment of the non-clinical data before going to the clinical development. There is scientific advice, and that's been proposed or expected. But even in the scientific advice, it's advice, and there's no data assessment per se within the construct here. So with that regard, and not looking to change how that happens, but it might be beneficial to put into these guidance language that does exist in other guidance about the recommendation to seek scientific advice, particularly to present the non-clinical and pharmacologic characterization in presenting a clinical program. And I'd even advocate that maybe the scientific advice committee that's evaluating the program would ask questions about the non-clinical, since that is to inform the type of clinical study needed and the totality of evidence being generated. And maybe even stronger language that, in particular, if there are non-clinical characterization aspects that are identified to be different from the reference product, maybe even stronger language to discuss those with the agency for scientific advice prior to moving into a clinical development phase. So the second point is really talking about the clinical development phase now. And we understand this is a general guidance and it's not class specific, but more specificity as to what is the thought process would be beneficial to the sponsors as well as the healthcare community, just to understand, again, what is the thought process or how are these populations going to be assessed. So there are multiple concepts and aspects of a clinical development program. The first is the pharmacokinetics. Here, it's important to specify that the population studied for pharmacokinetics should be sensitive to detect a difference if one exists. And hence, this could be healthy volunteers or it could be patients, depending on the actual molecule being studied. We do know and have to appreciate that PK does not inform about in vivo efficacy. It is strictly that of circulatory time of the molecule to affect its target. So absorption and clearance, and that's really it. So it's limited information, certainly no information on efficacy as a PK assessment itself. If in patients, you might get some hints of efficacy depending on the study design, and limited safety or immunogenicity, again, depending on the study design and the patient population. So we believe that equivalent PK demonstration relevant to the reference product is necessary, but it's not sufficient to make any assessments of clinical equivalence, whether it's efficacy nor safety. So we would also do agree with the guidance for pre-specifying and justifying the PK equivalence margins, but we do recommend a statement that PK is necessary but not sufficient for demonstrating similarity clinically. So we have a comment about PD or pharmacodynamics. We recommend the additional discussion explaining the limitations and providing some criteria for using PD markers or potentially multiple PD markers when they're not an accepted surrogate. So we're not saying that they all have to be an accepted surrogate. We know that there are limited numbers of actual validated surrogate markers. But what is the criteria for a marker to be relevant? We think it has to be clinically relevant and has to inform the mechanism of action. An example might be tumor response in oncology, and it might be beneficial to put such examples into a guidance just to know what the thinking is or to illustrate what the thought process is. The PD markers must be sensitive to inform clinical efficacy if it's trying to be used in lieu of an efficacy study. And to inform, it means there's some dose-response relationship and understanding of the PD marker with that of clinical efficacy. Further, it should have some predicted value with regards to clinical efficacy. So we recommend adding a statement about the PD markers that they may be sufficient to conclude, and here instead of clinical comparability, it probably should say clinical efficacy in some circumstances. And in those circumstances, there's still a need to assess safety and immunogenicity, because at that point, that's not been determined. So specific now to the clinical study looking at efficacy and safety. What is a sensitive population or model is a specific question. And I think it's important actually to consider both of these at the same time, because one can have a sensitive population with a non-sensitive endpoint, or you might have a sensitive endpoint in the wrong population. So it really only matters when you get it right and you get the sensitive endpoint being evaluated in a sensitive population. Of course, sensitive is up for definition or in the eye of the reviewer, what makes something sensitive. So understanding that, what criteria, what the thought process is for assessing sensitivity, could it be helpful? It should be used to confirm the comparable efficacy. And in doing so, the equivalence margin is important to pre-specify. The efficacy equivalence margin depends on the effect size of the reference product, as well as the sensitivity of the study model, that being the population and that endpoint. I think it should be minimally required that the equivalence margin rules out no effect or the placebo effect. So there should be some understanding of constancy, similar to the primary principles of non-inferiority. So we should make sure there's some constancy, and we've ruled out no effect at a bare minimum. But more than that would be what is the acceptable clinically meaningful difference based on those aspects of the reference product and the efficacy of the product in the studied population. So the study population needs to inform also about the safety. And here I'm looking or discussing more about the events of interest that are known for the reference product. These should be clearly assessed, and the population should be able to inform about these events of interest of the reference product. And obviously look for any potential overt new biosimilar specific findings. They're not expected, but they have been seen already in clinical trials of other biosimilars. So even if it's expected to be a rare event, they should be evaluated to make sure they are indeed rare, as well as no new surprise findings before a commercial authorization is given. So we'd recommend some more specificity as to the definition of a sensitive population in the endpoint, or at least the thought process as to how it's being assessed. And again, can include an example of what, and if it's too hard to put examples into guidance, we understand that. Maybe a separate Q&A type document could be of value to further illustrate the definitions and how to interpret the words in the guidance. So similar thoughts on immunogenicity, to request a little bit more specific expectations as to the assessment of immunogenicity during the clinical development phase of biosimilars, and understanding how you think about, as an agency, the duration or the number of exposures that are appropriate for assessing immunogenicity. So that kind of clarity could be beneficial, certainly to sponsors, as well as the larger community. Pre-approval immunogenicity detection depends on the characteristics of the reference product, but also the population studied. For example, whether they're immune suppressed during the study, whether they're receiving chemotherapy, obviously affects the signal detection for immunogenicity and anti-drug antibody formation. So further to recommending clarity, I think earlier there's been discussion about an expectation of at least 12 months immunogenicity for chronically used molecules, and there's been some other discussion about maybe shorter duration for molecules even when they're potentially chronically going to be used. So again, understanding what is that, what determines that, and examples. What's the thought process? Is it that you've established the the immunogenicity plateau, and once you're on the plateau, you followed long enough? That would be an important concept to know. If that's what's needed, we would understand how to design the study and the duration necessary to show on the plateau. Or if there's linearly increased anti-drug antibodies with increased exposure over time, you don't go on for infinity, but what's the point in time for which you've established the same rate of increased immunogenicity with increased exposure? And finally, if there's lower immunogenicity, as we discussed this morning, we agree that that does not preclude biosimilarity. The observation of lower immunogenicity needs to be understood and explained, as opposed to just a surprise finding. It should be well understood. And in that circumstance, we agree with the agency with the efficacy assessments being by subgroups for those who have antibodies and those who do not, to then best assess similarity. So now, big topic of extrapolation of indications. And here I think it's important to take those concepts we just had on sensitivity of population, sensitivity of endpoint, immunogenicity, but it's now a bigger question. Does that study not just inform the indication that was actually studied, but how well does it inform all the other indications being sought? So we need to make sure we have understanding of the mechanism of action, and with that also is the pathophysiology. That was discussed just prior. Where are the receptors, the density of receptors, but is the disease the same disease, not just the mechanism of the action of the actual intervention? So is the clinical study that's been conducted, is it sensitive to confirm equivalent efficacy without a study in the other indications? Is there a different indication for which, if the molecule isn't as sensitive, you would have diminished efficacy? And is it fair to put patients on that drug in a post-marketing or an approved setting? Similar to safety and immunogenicity, does the studied population really inform the safety or immunogenicity of the indications sought through extrapolation? For example, if you study in a population receiving immune modulators, methotrexate or chemotherapy, and then there's indications that there is no immune suppression, I would challenge that we probably do not really understand the true immunogenicity as a comparison of the two molecules in a non-immune compromised state. So that needs to be considered for extrapolation. So we'd recommend specific language to the appropriate pre-approval requirements for efficacy, safety, and immunogenicity in order to extrapolate, and how to approach and understand the populations studied for that extrapolation to inform pre-approval. So brief illustration and concept. This is a molecule that was discussed a couple of years ago in the MAB guidance conversation. And this is meant entirely to be illustrative of an anti-CD20 type of development program. If the clinical study was to be conducted in rheumatoid arthritis using ACR20 endpoint, and I know Dr. McCamish is going to discuss ACR20s, I'm sure, based on his slides, it's not very discriminatory with regards to efficacy for comparing two like molecules. It is discriminatory for assessing a molecule compared to placebo. So it probably informs about rheumatoid arthritis that there's no clinically meaningful difference, but I would challenge it's hard to inform about oncology in this case, where the oncology drug, for example, might be used in monotherapy. It might be used chronically. So what happens if you do an RA study for 6 months in this case? Well, that's one dose, 2 infusions 2 weeks apart, admitted, but it's one dose for 6 months. How well does that inform an oncology setting for which to be multiple doses, maybe used as monotherapy, maybe used as maintenance therapy for years? So I would be personally challenged to think that you have adequate information to extrapolate in that circumstance, and the clinical development program should provide the appropriate data ahead of time. So extrapolation, I think, is important. I'd even say critical to biosimilar developers. I would challenge that not many of us would be developing biosimilars if we did not think extrapolation was available, because the development cost would be prohibitive. However, it should not be a given. It still needs to be earned and justified. It needs to be based on scientific data, not just hopes, and supported by evidence, with efficacy, safety, and immunogenicity all considered. And this is to maintain confidence, not just amongst the company, the developer, or the regulator, but the confidence of the physicians and the patients. That's where it's going to be used and supported, and the longevity of the programs of biosimilars at large. We see the EU as the global leader in guidelines and the leader in the evaluation of biosimilars from an experience base. So maintaining clear scientific pre-approval requirements is critical for long-term success globally of biosimilars. So you have the added burden of being the leader, and so we need to get it right. So we recommend more clarity in the guidelines on the key aspects, accompanied by the full transparency in the scientific decision-making. So lastly, overall approach for biosimilars, resolving key efficacy or safety questions should not be a post-approval exercise. We all benefit from increased specificity of standards, even for the case-by-case assessment for which all these molecules really are. But potentially having the same rapporteur and co-rapporteur evaluate the molecules for the same reference product adds value, it adds consistency, and facilitates management of any safety issues in the post-approval setting. Patient and physician confidence are key to biosimilar uptake. We need to support them and give them the evidence to desire to use biosimilars. So good communication of the science behind the approval, combined with the appropriate post-approval data on immunogenicity, for example, in extrapolated indications or rare safety events as appropriate, the ability to rapidly detect signals for individual products will benefit both originator and biosimilar manufacturers, and ultimately will benefit the patients themselves. Thank you.